Okay, great. I think we're going to start this session. And so we have three wonderful speakers lined up today, the first of whom is Niccolò Fusi, who is a researcher at Microsoft Research New England, which is in Cambridge, and he works at the intersection of machine learning and computational biology. His focus is on the development of new statistical and computational methods to better understand the genetic and environmental causes of complex diseases. And in machine learning, his main interest is in the development of scalable inference methods for Bayesian non-parametric models. He received his PhD in computer science from the University of Sheffield, working with Neil Lawrence, and he received his bachelor and master's in theoretical computer science from the University of Milan. And today he's going to tell us about his work in CRISPR gene editing.
Thank you, Jennifer. Thanks for coming. So, as Jennifer just said, today I'm going to talk about machine learning for CRISPR gene editing. And I want to start by acknowledging how incredible a place Cambridge, Massachusetts is. And in particular, this is a map of Kendall Square, where the Microsoft Research Office is. And if you look all around, you see a lot of pharmaceutical companies, top world-class research institutes like the Broad Institute, MIT, Harvard, various IT companies. And it's a very exciting place to be. And it's part of the reason why the projects I'm going to talk about today even just started. And in particular, that's how we got to meet our great collaborators also here in the audience, John Dench at the Broad Institute of MIT and Harvard, and this joint work also with Jennifer Lesgard at Microsoft Research. So probably in the past 2 years, if not maybe the last year, you've heard a lot about gene editing. It's prominently featured in pretty much a wide spectrum of the press, from more technical journals to economics journals to the New York Times to CNN. Everybody is recognizing its potential to revolutionize medicine, agriculture, drug development. Some publications slightly overdid it. Wired called it the Genesis engine, saying it would end anger, pollution, cure all diseases, and it's the end of life as we know it, which didn't go down particularly well on Twitter. So quite a few people made quite a lot of fun of this. Anyway, despite the hype and under-hype cycle, it's showing a lot of promise for translational medicine. So in proof of principle applications, people have used it to remove a CCR5 receptor used by HIV, correct defects associated with cystic fibrosis, and correct muscular dystrophy gene to produce mice cured of the disease, just to name a few recent applications. The point is that this is the analogy that I usually use when talking about CRISPR. What the press and what the people, like I personally, want is the equivalent of a Tesla Model S. What we really have is a Ford Model T. As the Ford Model T, this technology has the potential to democratize access to gene editing, make it easier and more accurate. But there are a lot of the system, there are a lot of parts of the system that we need to improve to go from this to that. And today we are going to change the tires maybe and put a better steering wheel on it. So in particular, I'm going to focus on two aspects of this system that we've worked on. And one is how to better characterize and predict on-target efficiency. And that is the problem of, given that I want to edit a position in the genome, how do I make sure that that edit actually is likely to succeed or actually succeeds? Not all edits work. And the second problem that we're going to tackle is how to estimate and reduce, and possibly reduce the off-target effects. And this means, given that I want to edit a specific location in DNA, how do I make sure that the edit doesn't do something unintended somewhere else, causing havoc and having unintended consequences? And we call these two projects Azimuth and Elevation. And basically, the names come from ballistics, what you use with rockets into space. You have a target you want to edit, and we get you there sometimes with high probability. So given these two problems, there are two possible solution paths. One is to improve the lab protocols and the biology behind the system, and people are working on it, and they're having quite some success. But also, and this is what we are focusing on, one can apply machine learning to prioritize experiments and to guide experimental choices. So just as a very quick history of CRISPR, it's one of the great stories and great successes of basic research. It started with basically started with a community of people studying bacteria, and as it often happens, people not working maybe in human diseases get a bit less funding, and so they were relatively isolated and not super well funded. And then through a series of events, including a yogurt company discovering that that some cultures of bacteria used in yogurts were surviving viral infection and some others weren't, people had discovered that this CRISPR system has a naturally occurring bacterial immune defense mechanism. And a few years later, people actually realized that one can co-opt this bacterial immune system to perform generic editing in any organism. And of course, this is now, as I said, having implications in human diseases, is speeding up drug discovery and development, and has the potential to develop better crops. So I don't want to embarrass myself because John is in the audience and he knows the biology a lot better than I do. So I'm going to talk about bookshelves here. I'm not going to talk about a lot of biology. The idea, my understanding of the system, or my metaphor for the system, is that bacteria have developed some sort of scrapbook of viral memories, of memories of viral infections. And CRISPR stands for clustered regular interspaced short palindromic repeats. So it's a place in the genome where you have some structure that is composed of palindromes that are equally spaced. And the way I interpret those is as kind of like the frame of a bookshelf. And then these viral memories are kind of the books you put in, and they are memories of previous infections. And the idea is that a bacteria puts one of these books into the bookshelf when it gets infected. And if the same virus tries to infect the bacteria again, then there is some template matching going on. So it looks at the books in the bookshelf, sees the bacterial DNA, the viral DNA, and then recruits these enzymes to cut and disable the invading DNA. And so basically, it's a kind of like a pattern matching system that recruits some giant molecular scissors. And people have realized that if you design a book to put in the bookshelf in just the right way, you can actually co-opt the system to perform edits in humans too, or any organism for that matter. And so the idea is you want to design one of these guides or viral memories such that they match some part of the DNA you want to edit. And so I'm going to start with the on-target problem, so how to improve make sure the edits actually happen. And later on, I'm going to talk about the off-target. From the machine learning point of view, this problem is very interesting, but there are not a lot of moving parts. So I hope to get through it relatively quickly. And then there is a more complex scenario to analyze for the off-target problem. So if you remember, I said given that you can design one of these synthetic memories or one of these books to edit a gene, well, the problem is that you can have, for an edit for any given gene, you can select many different books, or you can generate many different synthetic viral memories. And not all of them work. Some of them don't work at all. Some of them work better than others. And so the idea is that we want to use machine learning to figure out, without performing an experiment, before we perform an experiment, whether a guide is going to work well for a given gene or not. And so the idea is that you can gather data, trying every possible RNA guide, and then verifying in a small subset of genes for which you can actually measure whether the gene edit was successful or not. You can try and see whether that was effective or not. And then the idea is that with machine learning, we want to learn this function that maps from the guide to the effectiveness, such that we are able to predict effectiveness. And from the machine learning point of view, you can see that as combining some input features, some properties of the guide that we can extract, and the measure get efficiency, which you can do in a lab setting for a small set of genes, through a model. And once you merge these three things together, you have a predictive model that you can apply to new data. So basically, the contributions that we made to this problem are three. First of all, we investigated and incorporated the richer features of the RNA guide. Then we removed some information bottlenecks from the supervisory signal, which was this success or not of the gene editing experiment. And we explored richer model classes. In particular, we looked at nonlinear models, whereas most of the models published in the literature, the few models published in the literature were largely linear. So this is this is how one of those blocks, one of those guides I've been talking about looks like. It's just a stretch. It's just a sequence of letters, a stretch of DNA. And the idea is that you want to go from some letters, which a machine learning algorithm wouldn't be able necessarily to understand, to some featureization of it. So some vector that describe what's in this sequence in a way that an algorithm can understand. And for instance, you can incorporate features such as, is there an A in position 1, or is there a T in position 14? And those are maybe some of the more obvious ones. We also use some of our knowledge in the area to incorporate less obvious ones. And when we were really struggling, we did JAJ, which stands for Just Ask John. And we did some more fancy featureization there. So I'm not going to go through all the features that we included, but some of them are easy, as I said, relatively easy to understand. Others capture complex things going on in the cell. One in particular is the melting temperature of the guide. You can interpret that as a proxy to the amount of energy needed by the system to open up the DNA at the location you want to edit. So there is this little animation that maybe shows it in more detail. The system, before you can perform the cut or the edit, has to bind to the target region. And so it has to bind and open up and unzip. And there is an amount of energy required to do that. And a lot of that, or some of that, is captured by this sequence, which is the guide you design. And so basically, we compared how much better the model becomes when you include these additional features as opposed to the features that are more or less more obvious to include. And as you can see, in terms of speedrun correlation, it consistently outperforms the more basic set of features across different data sets. I'm not going to explain much more about the data sets. Finally, we did two more things, which is to remove information bottlenecks. And that allowed us to use more complicated models. So in particular, one easy way to see where an information bottleneck could be is that the experimental method used to generate the supervisory signal, so whether a guide was successful at an edit or not. So that supervisory signal was on a continuous scale, and a lot of the previous approaches were just discretizing it in did that work or not. But the notion of working versus not working was not immediately clear. It was very gene-specific. And so it introduced some bias and some bottleneck in the model. So we removed that. We switched to regression after some pre-processing of the data. And in particular, we switched to nonlinear models, which allowed us to capture the more complex interactions between different features. And in particular, we settled on using gradient-boosted regression trees. They're a really popular technique in machine learning. There is not much to say. They're basically an ensemble of weak predictors. So they're a combination of multiple models that individually wouldn't do very well on this data, or most data. But when you combine them and chain them together, they outperform usually any individual simple model. And another attractive feature is that they can easily handle non-homogeneous data, which is what we have here, because we have a mixture of categorical variables, continuous variables, and so on. And the red bar is the final model, and the best performing model is like the shaded blue. And basically, across all data sets, we improve the state of the art in a statistically significant way. So Since publication, this paper was published in Nature Biotechnology in January of this year. Independent studies have verified on independent data sets that this is indeed the state of the art on target prediction. It has been adopted by startups and several research institutes and universities. The Azure machine learning service that serves the prediction, which is basically a cloud service that people can use to access our model, has been answering roughly 1,000 requests a day, so roughly 200,000 requests since it went online, and with usage doubling every 3 months. And the web service hosted by the Broad Institute is also answering roughly 300 requests a day. And we also made the source code available and a Python package available, and we got over 1,000 open source software downloads. So the second part of the project is the elimination or reduction of off-target effects, and this is what we call elevation. And this is a much more challenging and interesting project in terms of, at least on the machine learning side. And that's because it's the data is very peculiar. We mostly have data for one mismatch. So let's say that you have an intended target that you want to edit. And let's say that you have an error, somewhere else in the genome, there exists a region that has the same sequence except for one letter. That's what we call a one mismatch. And it turns out that there are a few targets like that. So let's say we're going to see in the next slide, there are roughly, for a string of 23 letters, there are roughly 69 sites that are the same, just with one letter different. The problem is that there can be more than one mismatch. So there can be 6, 7, 8, and the number of sites that have the same sequence but with 6 mismatches grows exponentially. And the data that we have is mostly for one mismatch. And we have a tiny, like a few samples for multiple mismatches. And the problem is that you need to scan genome-wide to identify these mismatch regions. And it's a combinatorial explosion. And this slide shows it, I think, pretty clearly. This is just for one guide for one gene. So every gene can have hundreds of guides. So imagine multiplying these numbers by 300. So for one guide in one gene, you have 69 sites with one mismatch. But if you go up to 5, you have 8 million sites. And we have one full gene that has one mismatch data. So it's pretty exhaustively sampled. But then for everything else, we have very sparsely sampled data. So the previous state-of-the-art approach, CFD, which stands for cutting frequency determination, basically looks at the intended target, looks at a possible off-target, and featurizes it, saying the intended target at the T in position 8, the actual this mismatch target as a C in position 8, and it encodes it as a categorical feature, or equivalently a one-hot encoded feature. And then it computed the probability after measuring of target activities as present or not present. It computed the probability of having an off-target given that that mismatch was present. And when you have multiple mismatch data, it is taking the product of all of these probabilities, effectively assuming that there are conditional independencies in the data. So we generalize this approach, switching once more from classification to regression to make use of this rich supervisor information. And we augmented the feature space to incorporate more information, giving more context to the model about what are the properties of the guide, of the mismatch, and the region being targeted. And also we used nonlinear regression models, again, boosted regression trees. And we used a second model to refine the predictions of the previous one. So this is like a cascade of 2 models, where the first model does all these 3 things with the purpose of using the better sample one mismatch data. And then there is a second model that takes the predictions from the first model and tries to relax the independence assumption. So it still looks at the product of the predictions, like CFD, but it looks at other things to try and mitigate violations of those assumptions. So the first model, as I said, it's a nonlinear model trained exclusively on data with one mismatch per site. And we have a lot of data for this problem, relatively a lot of data, because it's all for one gene. But basically, this is a complex model that can capture interactions, but can only compute predictions for one mismatch at a time. And then the second model is a linear model that is trained on the very little multi-mismatch data that we have. And it's relatively simple. It's linear, but that's all we could afford given the data that we have. And it's trained both on individual predictions from the first model and some function of those, such as the product. And so this is still work in progress very much, but our early results show that it performs the previous approach by a statistically significant margin in the main data set we look at, but also across other 4 out of 5 data sets that have been published in the literature. And what's interesting is also looking at how much the second model steps in to check for assumptions that were not respected. So basically, this is the weight put by the second model on the various features that we give. So of course, the most important feature is the product feature, meaning that the assumptions made by CFD were not unreasonable. But then there are all these other features that step in to correct these assumptions when they're not met. And for instance, predictions of a mismatch in position 1 turn out to be important as prediction of mismatches in position 14 or the number of mismatches. So putting it all together means we have to perform this kind of work. You need to apply this kind of model. You need to scan genome-wide for every single guide you want to design. So you need to. The on-target activity is easy. You give the guide to our predictive model. It returns a number. To predict the off-target activity, it needs to look everywhere else in the genome for strings that have 1 letter change, or 2 letters, or 3 letters, or 4 letters. So that's very computationally expensive, which is why we are developing a cloud prediction service that has everything pre-computed for the human genome and possibly other genomes. We are also going to release open source code specifically for this project. And together with Michael Weinstein at UCLA, we're also developing a framework to efficiently search genome-wide for mismatches and compute predictions both for the on-target efficacy using azimuth and the off-target predictions using elevation. And I would like to conclude by thanking again John Dench at Broad Institute at MIT and Harvard, and Jennifer at Microsoft Research. Thank you.
Great. I think we have about 5 minutes for questions. And there's someone in the back with a mic, so just put up your hand and she'll pop by.
On the first model? Like the final one? This one?
So is L1 regression working better on first data set?
Yeah. It's in this case, the difference was not statistically significant, which is why it doesn't have the star. It's the amount of data here is very relatively scarce. So there is a bit of difference between L1 and nonlinear models, but I don't think it's statistically significant. Well, we know that it's not statistically significant.
And how did you pick the final model? Did you search using other data to try to figure out which model was working better?
We cross-validated across so we had 2 data sets. We had a data set that was published in 2014 or 2015, I don't remember, 2015, I think. And then we had a new data set that was generated with a slightly different, well, with a different technology. So we did a lot of the cross-validation on the first, but then we also combined them and also cross-validated as the combination of the 2.
And as he showed, there's another paper that just came out that independently confirms that this is the state-of-the-art model.
Yeah.
I was just wondering if you didn't use the data. I mean, you tried to use it for
We tried as much as possible to not touch the data too much. So to not basically overfit to whatever data set we had. I think up here in the front, there is a mic problem.
I think in the interest of time, we should probably, do you want to take one more? Or?
Well,
Let's do one more, and then we'll switch to the next speaker.
So for the off-target effect, have you looked at using any aligner technology? For example, looking at using genome like aligners or mappers?
To find them?
Yeah.
You mean
so
to find the sequences to score?
Right. So for example, SNAP builds a hash table of all the k-mers in the human genome, and you can search that very efficiently.
Yes, that's something that people, well, not specific with SNAP, but that people have used some aligners before to do this. We have a collaborator at UCLA who has developed his own tool based on a seed and extend algorithm that basically is well suited for their HPC cluster, and we just use that. That's it.
Okay, great. Let's thank the speaker, and then we'll move on to the next one.
You can take another mic. Yeah. Just press next.
She should just press next.
Yeah.
Great. It's my real pleasure to have Donna here, who is a friend and hopefully one day a collaborator. So Donna develops computational methods that integrate diverse high-throughput data to provide a holistic systems-level view of molecular networks. Currently, she has two key focuses: developing computational methods to interpret single-cell data and understand cellular heterogeneity, modeling how genetic and epigenetic variation alters regulatory network function and subsequently phenotype in health and disease. And this path has led her to explore how systems biology approaches can be used to personalize cancer care. Donna is the recipient of the Burroughs Wellcome Fund Career Award, NIH Director's New Innovator Award, NSF Career Award, Stand Up to Cancer Innovative Research Grant, and a Packard Fellow in Science and Engineering. And she gives wonderful talks, so here we go.
Okay, thanks for inviting me. And I didn't quite know who the audience would be, so. So usually we're in the problem as researchers to find excuses of why our problem is important and why we're doing important things. But in some of the things I do, I've had my excuse given to me. So the president has defined that this is an important project. And we really are at a wonderful time where we as a machine learning community, we as computational scientists could have really meaningful impact on health, on saving lives, and making people's lives much better, and that is using data and data measured from people and health in order to personalize healthcare and improve it, because healthcare has become really data-driven. Now, this has caused a lot of people to work very hard on this problem. Certainly when the president says, this is important and I'm going to fund it very deeply, people jump on the bandwagon. And even Microsoft Research has been doing great things along these lines. Now, I'm going to, say that I don't think everyone's going in the right way. And the vision is, okay, we go from a DNA directly to phenotype. We're going to be able to figure out everything and all the differences and all the personalization based on what's written in each and every one of our DNA code, or going to sort of a more genetic viewpoint. We have the DNA, we have cases, we have controls, and we can directly predict from the DNA what's going to happen. And I think that's only going to take us so far. And that's because we found out that actually most of the variants that cause diseases, most of the places that are different between me and many of you in the audience are not in the genes themselves, but directly in the regulatory regions. So what most of the genetic variation, both in healthy traits and in disease traits, code for is telling a gene to be expressed at a different level, at a different time, and at a different place. So in order to really match between the DNA and phenotypes, such as disease, we're going to have to understand this intermediate of regulatory networks and gene expression. What makes this even more complicated is this is not the same thing for the whole body. So most diseases are caused because a certain gene is misexpressed in a certain, often rare cell types. In many cases, these cell types were only discovered a few years ago. So just finding out, okay, there's an association here. Here's a region well outside a gene that is associated with disease doesn't tell us why. Doesn't tell us, well, this is causing a disease because in this very specific cell type, something is going wrong. So we need to look at our cell types. And we have many cell types. And each of these cell types have a different regulatory program. And each of these cell types have a different piece of the DNA that's regulating them. So a piece of regulatory DNA is specific often to one cell type and not another. So when we actually find some association in the DNA, we have to figure out what gene, what cell, when, why, and how. And one of the problems here is that we're skipping a fundamental thing. Genes are fundamental things. We've mapped a genome. We have a genome. But what we don't have is our cell types. You ask someone how many cell types we have in our body, they'll throw the number 400. Then you ask an immunologist, how many immunology cells do we have? And that immunologist will say, we have 400 immunological cell types. And you ask a T-cell biologist, how many T-cell types do we have? And they'll also give you the number 400. And in truth is, we've mapped our genes, but we only know a fraction of the different cell types in our body. And actually, many diseases are caused by rare cell types, uncharacterized cell types that are 1 in 100, 1 in 1,000, 1 in 10,000 cells. So in order to really do precision medicine, in order to use these genes and figure out what's going on, we first have to map our cell types. And to do this, we need single-cell technologies. We need to actually measure cells at single-cell resolution and figure out, well, what do we have in our 40 trillion cells in our body, and how do they disassociate into different groups? And the fun thing is there's a lot of technologies that gets at it. There is CyTOF, which can measure 45 proteins simultaneously in millions of individual cells. We have single-cell RNA-seq that can go genome-wide in thousands and tens of thousands, and some new spatial technologies coming up. And this is a wonderful playground. As someone who was dealing with a lot of genomics and was dealing with the problem of 20,000 genes or 3 million SNPs and 100 samples, finally you are at 40 dimensions and 16 million cells. Yay. But you have to try and begin understanding this new data type, this new playground of single-cell data. And so first of all, what is a phenotype? A phenotype is basically a cell and where it's located in the cell. So we measured the space. We have 45 proteins. We have a gene expression vector of the gene. It creates this high-dimensional space, and cell types, cell phenotypes, cell states create these densities in the space. And now we actually have to find these densities. So the first thing, picture is worth 1,000 words. We found that T-SNE nonlinear dimensionality reduction really helps us visualize this data so that every single cell type falls in a different region in the map. That's because these cell types actually fall on manifolds in this high-dimensional space, and now T-SNE nonlinear dimensionality reduction is broadly used across the single-cell field to visualize data. And here, going into one more dimension, I'm looking now at 3 dimensions. This is the exact same data. You could really see that this data creates complex, concave, nonlinear manifolds. And these are often modeled as sort of nice, beautiful Gaussian or ellipsoid balls. And you can look at it, decide if that's a good model for this data, ellipsoids. It's complex and it's beautiful. And we actually try and capture this data, they sort of map the shape, the geometries that we see in cell type space in a graph structure. The thing is that distances, long distances in this high dimensional space are very misleading. And most geometries give weight for these long distances, which we don't trust. And this graph-based representation, where every cell is given a node in the graph, and we connect very similar cells one to another, puts focus on the measure that we trust, that when cells are very, very similar to each other, then we trust that they're phenotypically close. So we take this high-dimensional space, convert it into a graph, where the nodes are cells and connected to their similar neighbors, and then we can actually use this graph as a mesh to model this high-dimensional space and work with this lower dimensionalist object, and we can do a lot of things. Here, simply, we try and find the cell types. We try and find these densities. It's a very simple algorithm. We do one more step to make it modular, which is to use the Jacquard metric to say I'm going to basically do an intersection of how many neighbors I share. This helps overcome the fact that we have sometimes 5 orders of magnitude difference in sizes of cell types. And this sort of cleans up noise from very small and tiny and important cell types, such as stem cells. And this magically works once you use your favorite community detection algorithm. Here, what you want is speed. This is a little snippet from a 16 million cell graph, so you want it to run quick enough. But really, once you do the Jacquard, you get that modularity that gives it pretty nicely. And again, when we know the answer, it works well. It works better than state-of-the-art algorithms. It works really fast. And we can now use it to explore new cell types. We've used it to explore cancer heterogeneity. And what I really want to use it for is to understand cancer immunotherapy. So, we talked about disease in general, and I focus a lot on cancer specifically. And again, cancer genomics and precision cancer medicine is something that the machine learning community has been deeply engaged in. But I, think a little bit also focused on the wrong problem, because everyone is focused on targeted therapies, and they're great because there's a lot of data for them. We have the TCGA, we have all these cell line screening data sets. But really, immunotherapy, science breakthrough of the year 2013, is promising to be the most promising avenue out there. For example, for metastatic melanoma, recently uncurable, now we can give 40% of the patients durable responses. Not give you a drug that will give you a release for 6 months, like the targeted therapy, but give a drug that will give you a relief for 5, 6 years and counting. And this is incredible. It's been working for a number of uncurable, really bad, hopeless case cancers, such as glioblastoma, lung cancer, bladder cancer. It's really showing a lot of promise, but the problem is it's working only for a small fraction of the cases. When it works, it does miracles, but it doesn't work for everyone. So our goal as a machine learning community should be to figure out why and how we can personalize this and bring immunotherapy to all cancer patients. The reason immunotherapy is so good and targeted therapy is so bad is because our immune system is smart. Our immune system is evolving. Cancer is a rapidly evolving disease. You give it any drug in the world that's static, and cancer will figure a way around it. But if you activate the immune system, our own immune system, against the cancer, that too is evolving, and then the immune system gets activated and can evolve and track the cancer and get at it. So we really need big data approaches to understand immunotherapy. But immunotherapy is not a single simple drug. It's not a small chemical that targets something. It's actually turning on a whole system. It's turning the on switch on a system of interacting cells and cell types, like I showed you earlier, and many of them haven't been characterized. We know a lot about the cell types in our blood. Those have been extensively studied. Our blood, our bone marrow, and health. But tissue-resident cells, cells that are in the lung, cells that are in the breast, cells that are in the bladder, cells that are in cancerous tissues, these haven't been characterized at all. We're using our imagination of what healthy cells look like to think about what they're doing inside the cancer. So the first thing we really need to do is build an atlas of the system that we are activating against the cancer. We don't even understand a drug. And for this, we're going to basically measure lots and lots and lots of single cells coming directly from patients. So we're going to sort out the immune cells of patients, shake the tumor, and get out these immune cells and measure them directly. We're going to measure them with this in-drop technology. This in-drop technology right here basically takes these cells. You see this tiny thing is a cell. It's encapsulated with a bead. And now we can take all the RNA in that cell for about 20,000 cells per sample and tag that molecule with a specific identifier for the molecule. This is the molecule we tagged, and a cell. This is the cell it came from, using these primers on the bead. And now I can, in a single experiment, and $500, tag all these cells. Of course, sequencing them is a little bit more than $500. And we started collecting these cohorts. And we started with breast cancer because it's one of these cancers which really works sometimes and usually not. So it's a good place to start to understand when it works and when it doesn't. And we're building an immune tissue atlas of the immune breast cancer system. Right now we have more than 100,000 cells measured from 10 different tumors. And I'm going to focus on 4 here because that's what I have the slides ready for. And the first thing you see, every dot here is a single cell plotted with the t-SNE we saw earlier that's supposed to find the structure and the shape of the population. Every dot is colored with a different tumor. So these are the 4 different patients that are in this picture. And you could see these huge tumor-specific regions. This stuff right here is something called myeloid cells. They're the innate immune pathway. They basically are our older immune system that just attacks what seems to be wrong. And these are the lymphoid cells. This is the sort of adaptive immune system that's capable of recognizing specific things that it saw in its past and attacking in a more targeted way. This is where the T cells go. And so here, looking at this big purple blob from patient BO, this, is very different from the other T cells. It has very activated metabolism. Here, this blob of myeloid cells right here in patient CA has more interferon. It's more like inflammatory than the others. So I can say, wow, each patient's immune system is different. We have individual, unique, precision, personalized immune systems for each patient. I can characterize these differences. I can put this up in Science, the journal Science, and I can guarantee you it will get accepted. Except I'm slightly a more careful scientist than that, and I'm wondering, is it truly patient differences or is it batches? Because all these experiments are hard, and while I describe them and explain them in a sort of hand-wavy way, look at how beautiful and simple it works. Every little step in the way can create differences and problems that might be causing these differences. So the question is this really different biology that I can base some health choices on? Or is it the process in which I generated this data? And the problem is that this data is very messy. It's very attractive. Yes, I have 100,000 cells directly out of cancer patients, and I measured their expression at single cell resolution. And now I can imagine in my beautiful, wishful thinking mind that every row here is a different gene, and every column is a different cell, and I have for each cell all the genes accurately and correctly measured, and I can now use t-SNE or clustering or every other thing I can do to find groups, subsets, and figure out what's going on. In reality, single-cell RNA-seq is filled and rife with technical artifacts, and one of the biggest ones is dropout. This dropout means that I only assay 2% of the cells inside a cell. And I could say, okay, I can build some nice model and I can assume this dropout is equivalent. First of all, as machine learning people, we have great methods to fill these matrix. We know how to fill missing data out. All these methods fail. Why do they fail? Because all our missing data methods assume that when we do have a positive entry, it's right. No, but the dropout is impacting everything. Even the entries that are positive are dropped out. We can't model everything differently. Every single cell has a different rate of dropout, and different types of cells have different types and flavors and distributions of artifacts. So what people do is try and normalize everything to equalize it. Let's make some assumption. Let's assume that all cells should have had the same amount of RNA to begin with. Let's assume that all the cells are sampled from one distribution of artifacts. But that's not true. We have different cell types in our immune system. They have different artifacts to the degree of an order of magnitude, and they have different efficiencies. So making these general assumptions fail. There are people that try and do spike-ins, but the spike-ins are shot into the sample after 80% of the variation has already occurred. So it only picks up 20% of the variation and costs you about 20 to 30% of your sequencing. So what are we going to try and do? We're actually going to try and use the shape of the data, the fact that the data has shape. We're going to use these nearest neighbor graphs and say, well, let's look at similar cells. If cells that are very, very similar to me are expressing something, I might be expressing it. It's never that simple, but we also have collinearities in our gene expression. We have modules of gene expression. So if I can't figure out just from the neighborhood, let's look at higher dimensions of collinearities and use the collinear structure of the data to figure out if it's expressing the gene or not. And of course, we're going to have to normalize it before we do anything else, because if we have different efficiency in these cells and one cell is sampled at a 10 or 100 times more efficiency, that's going to mess everything up if I don't correct and normalize for it. As I said, if I normalize everything, assuming that a cell like a macrophage, which has an order of magnitude or 2 more cells than a lymphocyte, is expressing the same thing, I'm going to mess everything up. So really, to normalize, I need to know the cell types. And my goal is to know the cell types. This is a classical computer science chicken or egg. I can't normalize without knowing the cell types, but my whole goal is to find these cell types so we can build this iterative algorithm that we're so good at doing in computational biology. Basically, we're going to try and learn the cell types, normalize according to these cell types, get better entries for our data matrix, and iteratively continue. We're going to model this expression as a mixture model, so each cell type has a different log normal distribution, and what we see is a mixture in one dimension. Of course, there's collinearity, so we have this high-dimensional mixture of log normals in high-dimensional space, and this is what we want to learn from the data. This is the nice model, assuming beautiful data, but our data is warped. Every single cell has this dropout, has this technical variant parameters that sort of warp it. So I can't learn this easily from the data because each and every cell in our data has been altered by these technical variants. So I'm going to use it based on a graphical model where I'm assuming, okay, I have a bunch of clusters that are expressing a value here out of a distribution. What I really see is an observed gene with cell-specific parameters that impact the entire cell that basically have warp the data. These cell-specific parameters are, again, specific for each cell from a distribution that's specific for each cell type. And again, to learn all this and infer all this, we're using hyperparameters and hyperpriors directly from the data. It's a hard Gibbs sampler to learn, so we need to learn hyperpriors from the data as powerfully as possible to limit the space so that, one, we'll find ourselves in a good solution regime, and two, this thing will run before my poor postdocs have to get a faculty position. We managed to get this to run. It's, again, very slow. I'm happy for any computer science help to make this run quicker, but we can get it to run on 150,000 cells with very good results. Again, we're doing this Gibbs sampler using a lot of tools that we know from the machine learning community to try and make this Gibbs sampler converge better and faster, and we have a lot more work to make it even better. But when we test it on theoretical data and drop things out, we recover what we dropped out. And again, the idea is we iteratively learn the parameters and learn the cell types, normalize the cell types according to these parameters, and in the end, we can impute what should have been a better cell matrix, recovering some of the data and recovering the structure of the clusters. As I said, we tested it on a lot of more synthetic data. We also tested it on some real data where we knew these answers. These cells were sequenced very deeply, and it's a subset of cells that had markers so that we really know, biologically speaking, what cell type there are. So this is sort of a gold standard data set, and our Biscuit algorithm did it very accurately. Now you could say, okay, this looks easy. So let's run some other algorithms. Doing it without the normalization really messed up. This is Seurat that's used most frequently for such data. It really messes things up. Here's the phenograph I showed you earlier. It actually gets a pretty bad F-score, but that's because it arbitrarily builds these 2 extra clusters, but at least it covers, you could see it gets the shape a little bit better. Now, when you normalize it using the standard normalization algorithms that people are using out there, you make it worse. You make it far worse. And this is because this normalization is assuming that all these cells should have the same number of transcripts, and we have a complex mixture of cell types in our data. This is real tissue data from patients. So now let's apply it to here. Remember, this is the patient that was going to give me the science paper of the personalized immune system of these cancer patients. But being careful, normalizing the data properly, distinguishing biological signal from artifact. This is the same data after BiSKIT. And you see that it's very well mixed. And you see that the data is far more structured. And we look at the clusters, you find all the known immune cell types that you were expecting to find very nicely split and clustered in the data. And this is not a biological audience, but we're finding a lot of biology here. We're finding a number of different myeloid-like cells that have different roles that we're looking into. Here in the T cell, we see an axis from here to here of exhaustion. Exhausted T cells are when they get too tired and stop working, so we can try and understand exhaustion. Whoops. Here. And so we're really looking into this data. So here I just showed you, hey guys, there are no differences. Actually, they are. If you dig into the covariate structure that we learned in order to fix the data, we see that the data is rich with covariates. And these covariates do change in a significant way. We could put p-values on them in a reliable way, in a robust way between the patients. What I'm showing you here are covariate matrices between molecules that are used by drug targets for immunotherapy. And you see that generally speaking in breast cancer, OX40 and GITAR seem to co-express on cells. These are different breaks. So when you have a cell that's expressing both GITAR and OX40, it means it's pressing on two breaks. If you cut one of those breaks, the OX40 is still there. So we need to target this with a double therapy, cut both those breaks. But you see this patient has a different combination of breaks, of ICOS and 4-1BB. So you see that there is personalization that we can get in what these immune systems are expressing, but again, in a much more refined way. And we need to clean up our data a lot more in order to get these refinements. So in summary, we've described to you a BiSKIT algorithm that begins to approach the complexity of cleaning up very valuable but very complex and messy single-cell RNA-seq data, which we're using to understand the tumor immune ecosystem and how we could use that to personalize cancer care and understand where the immunotherapy works and fails. More generally, now that we have that, I just showed you one project out of many that we're doing to personalize immunotherapy. We could look at these interactions with these populations, characterize them, understand what they're doing in the context of the tumor, and most importantly, how they change following drugs in both responders and in non-responders, integrating and using the power of machine learning to integrate different data types, additional data types, such as neoantigens, T-cell repertoire, and everything else to really bring precision medicine into the realm of immunotherapy rather than just precision medicine. So thanks for listening to me. Sandhya and Elham developed the BiSKIT algorithm, being able to get the tumor data and both technologically run the in-drop and analyze and process the data is joint work by Ambrose, Linus, and Josh, along with my collaborators at MSKCC, which I'm moving to in October. George, a wonderful clinician who really cares about his patients, and Kasia in Sasha's lab. And of course, thanks for the patients. These patients gave us their material likely to help future patients and not themselves. And thank you for your attention.
Great. Thanks again for a talk. And the mic is going to come from over there.
That was great. So when looking at the covariant structure that you showed there at the end, so there you honed in on CTLA4, which is obviously a known break, and some other known breaks. Is there enough information in the data if you didn't know where to look? Like, could you find ICOS coming out as, okay, here's another break we should be looking at relative to all the other genes that, might have some covariation but aren't obvious actual drug targets?
So there's a lot of covariate structure in the data, and we're analyzing this. This is unpublished work that we're now analyzing, and now, as I said, we've scaled it to 120,000 cells. What you're looking at is data from 12,000 cells right here. We have a measure for what's significant, and a lot of things come up as significant. Many of them involve unknown genes. we also are honing in on some things that are known from the immune system. We do get so much covariation structure that when you go in there completely blind and say, let me just get all the top pairs that are significant, you drown. This does come up, in the top 1,000 pairs, if you want to look at 1,000 pairs and stuff. So we are using a little bit of biological curation to bring up sets that are at least known to play some roles in immunity to begin to make sense of this data. I'm sure with time, once we make more sense, we can get more and more data-driven and unbiased. But we have looked unbiased across everything. We just get too many pairs, so we are not only looking at these, I just brought these up as anecdotes to try and guide, okay, how can we make sense? There's so much unknown in the system that you need to grasp on to a little bit of knowledge to begin interpreting. I need a lot of cells. This is very rare cells. Can you do CRISPR when I give you 130,000 cells?
Figure something out.
Okay. When you do CRISPR for 30,000 cells, we'll work together.
So
Gladly.
I was going to ask a question actually myself. Did you try variational inference at all to speed things up?
So we're working on variational inference, and the math here is hard. When David Bly saw what we did for this model and the things that we worked out, he told Sandhya, respect, you have my respect.
We have a world-famous expert in variational inference at the back.
I would
at
love
MIT,
to do this.
And you're both my friends, so maybe after
I would love to involve variation inference. And I said I'm everyone in this audience to make this better. I am a little bit concerned that I happen to know that variational inference isn't as accurate as Gibbs, and since this is so finicky and with so many unobserved variables, I'm a little bit afraid that making it a little bit rougher with variational inference will lose our accuracy. But I definitely am game to try. We've started working on it. We're not variational inference experts, and anyone who wants to do this with us, welcome.
Great. I think in the interest of time, we're actually going to have to cut short on questions. Sorry, but I think the speakers will all be around after if you want to ask. So let's let's thank Donna again. Great. So the last speaker of the session is Eran Segal, who I'm extremely excited to have here. I don't think I've actually heard him talk, but I've seen his work very recently, especially in the news a lot, and I'm very excited to hear this. So Eran is a professor at the Department of Computer Science and Applied Mathematics at the Weizmann Institute of Science, heading a lab with a multidisciplinary team of computational biologists and experimental scientists in the area of computational and systems biology. His group has extensive experience in machine learning, computational biology, probabilistic models, and analysis of heterogeneous high-throughput genomic data. His research focuses on nutrition, genetics, microbiome, and gene regulation and their effect on health and disease. He's published over 100 publications and received several awards, honors for his work, including the Overton Prize, which I believe Donna has also won, awarded annually by the International Society for Bioinformatics to one scientist for outstanding accomplishments in computational biology, and the Michael Bruno Award. He was recently elected as an EMBO member and as a member of the Young Israeli Academy of Science. And great, thanks, Eran.
Great. Am I on? Yeah. Thanks, Jennifer, for the invitation. Can we get the slides, maybe? Oh, I need to do that? Ah, okay. Here we go. Okay, yeah, so a few years ago I became interested in taking a scientific data-driven approach to the problem of nutrition. And there's several reasons for that. First is we all know of the huge rise in metabolic disease that we are witnessing today. So if you live in the United States today, there's over a 70% chance that you're either overweight, diabetic, or have non-alcoholic fatty liver disease. And I believe that changes that we've done in the past several decades to our nutrition have contributed to actually driving this disease epidemic. And I just want to note very briefly some of these changes. So about 40 years ago, the first one was not based on scientific evidence. We decided to change our macronutrient intake and reduce our intake of fat and instead increase that by necessity our intake of carbohydrates, which inevitably contributed to the rise in blood sugar levels and diabetes and obesity and so on. Another huge change is major increase in our intake of added sugar to food, when we all know the harmful effects of sugar. I don't know how many of you know the numbers, but the amount of added sugar that we consume in a single day today is more than in a single day is more than we've consumed over an entire year just 300 years ago. So this is one of the major nutritional changes that we've done. Another one was to add food additives to our food, most notably artificial sweeteners, which most of us consume on a daily basis in various diet soda drinks and other products. We had work several years ago showing that consumption of artificial sweeteners alters the composition and function of the gut bacteria in a way that can actually lead to the development of symptoms of diabetes. And finally, since the introduction of electricity about 150 years ago, we also began to engage in shift working activity. A few years ago, we also published work showing that even though our gut bacteria live in the dark, they also follow a rhythmic circadian cycle. And by shift working and even jet lagging, we actually alter that normal function of the gut bacteria such that when transferred into germ-free mice, can also cause symptoms of diabetes and weight gain. And so these and many others have actually contributed, in my view, directly to the rise in metabolic disease. And if it's changes in nutrition and lifestyle, then perhaps by restoring healthy nutrition and healthy lifestyle, we can actually reverse this epidemic. And this is what got me interested in it. And the approach we're taking is to try and combine various types of measurements, including our genetics, lifestyle, gut bacteria, and nutrition into this problem. And when we started out this project a few years ago, the first question that we asked ourselves is what marker of healthy nutrition should we follow such that we can really take a big data-driven approach to the problem. And we wanted this marker to obey several properties. We wanted it to be obviously relevant for weight management, but also relevant for metabolic disease, and not less importantly, also some marker that we could also measure easily and quantitatively, which is not something that's very easy to find in the field of nutrition. And thinking very hard about the problem, about this question when we started, this led us to focus on blood sugar levels, and more specifically, on changes in blood sugar levels after we eat a meal. This is what's called a postprandial or post-meal glucose response. And so, as you may know, our blood glucose levels mainly change after we eat, and we can actually follow the blood glucose levels and quantify then into a single measure using the area under the glucose curve in the 2 hours after eating a meal. We can quantify the effect that that meal had on our blood sugar levels. And this is important because spikes in blood glucose levels after you eat a meal actually cause secretion of insulin, whose job is to reduce the levels of blood sugar levels. But insulin, being an anabolic hormone, also signals our body to convert excess sugar into fat. And this is the primary way, actually, by which we gain weight. In addition, spikes in blood glucose levels can often lead to lower glucose levels below baseline levels, making us feel hungry and then behaviorally also eating more. So they're very critical for weight management. They've also been shown to be linked and be disease risk factors for many diseases, including diabetes, obesity, cardiovascular disease, and other metabolic disorders. And again, not least importantly, with recent technology due to the rise in diabetes, we can actually very easily now quantify and measure blood glucose levels even continuously in a person. And so you might think then the only thing that we need to do is to eat foods that don't spike our glucose levels. But that's easier said than done, because prior to our work, there's been some anecdotal studies, typically on 10 or 20 individuals on 1 or 2 different types of foods, showing that actually when you give different people the same food, they can actually have wildly different blood glucose responses. And this is just an example from one paper on 20 healthy individuals, where you can see some people given white bread having almost no change in their blood glucose levels, whereas in others we see large spikes. And when you see this data, that makes you realize that if you want diets that maintain normal blood glucose levels, they really have to be personally tailored to the individual. And that's really what led us to initiate this project that we called the Personalized Nutrition Project, in which we recruited, by now it's over 1,000 participants, and we measured their blood glucose response using continuous glucose monitors that measure for a full week blood glucose levels every 5 minutes. And by that, and people logging on a mobile app that we developed all of their food intake, we've logged over 50,000 meals, measured their blood glucose response, and have over 2 million blood glucose measurements. And then, anticipating that there would be this variability in people's glucose response, we wanted to see if we can understand the reasons for these variability. And so we've also obtained a very comprehensive profile from each participant, including medical background and food frequency questionnaires, various blood tests, body measures, and also a very comprehensive profile of both 16S and metagenomic profile of their gut bacteria population using DNA sequencing. And just to give you a glimpse on the data, here's a zoom in of the blood glucose levels measured across an entire week for one participant, where zooming in, you can see that blood glucose levels rise in this participant when he reported eating the different meals, and you can see that different meals actually have different effects on the blood sugar levels of this individual. Just in terms of our cohort, it was really representative of the adult Israeli population, with a relatively uniform distribution of age between 20 and 70, and a distribution of BMI with about 50% overweight, 20% with BMI above 30. And we purposely excluded diabetics, which gave us really a distribution also of kind of diabetes markers, which are at the normal range, representative of the adult Israeli population, about 20% in the pre-diabetic range. In the United States, it's about 37% of the adult population who are at the pre-diabetic range. And so what did we find? Well, the first question that we asked ourselves is going back to this variability between people. How variable are people in response to the same food? And as the most controlled way to look at that, actually in the study, we asked people to follow their normal dietary habits so that we could see how they respond to what they really eat. But in the morning, in order to standardize and compare across people, we provided the breakfast. They were very standard breakfast, always 50 grams of available carbohydrates, either bread with butter, or glucose, and each of these foods in duplicates. And so that allowed us to first compare what happens when the same person eats the same meal on different days. And here we found very reproducible responses, so that's good. The same person is quite reproducible to himself. But when we looked across people, we saw huge variation. And this is now on a scale that was never examined before. This is on a, in this case, 800 people scale. We now have it for over 1,000 people, where you can see that for all of these foods that we tested, we provided actually identical breakfast. You can see huge variation for each and every single meal. For each of these meals, you see some individuals that have very low responses, whereas other having very high responses. And the responses were reproducible within a participant. So here, for example, for bread, you can see an individual having almost no response and others having huge responses. And it wasn't also that just some individuals responded highly to anything that you give them. You could also identify clusters even when you normalize the response of each individual to himself. So you can see here that there's clusters of individuals. For example, in this cluster here, people respond most highly to glucose compared to the other foods, but in this cluster, they respond more highly to bread than to glucose. And you can see here even that some cases of participants that have exactly opposite responses, one participant responding more highly to glucose than to bread, and another participant having exactly the opposite responses. And so this really was very striking, because seeing this on such a large scale really tells you that diets that maintain normal blood glucose levels have to be personally tailored to the individual. And then with the very comprehensive profile that we collected, we then wanted to see whether anything in that data correlated with variability across people. And so we first just did a basic association type of analysis, And indeed, we found that across all the different foods that we tested, for all the different parameters, there were certain parameters that associated with variability in people's response to these different meals. And that was true both for blood measurements, body measurements, but also very interestingly for many different aspects of the gut bacterial composition, either the 16S composition analysis, the metagenomic analysis, or particular metabolic pathways that are enriched in the gut bacteria of some people. So for example, here's a certain group of genes. These are genes involved in ABC transporters that when they're enriched in the gut bacteria of people, that's associated with a higher response to all of the different test foods. Anecdotally and incidentally, this particular group of genes has also been, in another study, positively associated with consumption of a Western high-fat, high-sugar diet. So we found many of these different associations. And then, of course, the next interesting question that we wanted to ask is whether we can somehow integrate this very comprehensive profile into actually making a predictor of how people would personally respond to various different meals. And before I show you what we did, I want to show you what the state of the art today is. So state of the art is to use the amount of carbohydrates in the meal as a predictor. When you apply that to our 50,000 meals, you get actually a highly statistically significant correlation. It's a 0.38 correlation, but probably none of you would want to choose your meals based on the quality of this predictor. I will tell you, however, that many people actually make important clinical decisions based on this predictor, such as type 1 diabetics who actually need to determine the amount of dose of insulin to inject after a meal. This is the best algorithm that they have and that they actually use in real life today. And it's very well documented and known that this is not working very well. So what we did, and I don't need to explain it to this audience, is to devise a machine learning predictor. We actually use gradient boosted decision trees that were mentioned earlier today. And initially on the training cohort, we used it in a cross-validation, leave one out fashion. As our different features, we actually have thousands of different features from which we did some feature selection and ended up with a little bit over 130 different features. The features actually come from many different aspects of the data we collected. So there's some aspects, some features that are at the person level, and these include various features that we extracted from the gut bacteria involving composition and metabolic function of the bacteria. There's other features coming directly from features of the meal, so the various macro and micronutrients involved in the meal. Then there's also state or contextual features having to do with the time of day at which a meal is consumed, what was in the previous meal, taking the time of the meal relative to exercise, the intensity of the exercise, when people went to sleep. All of those came up automatically as being important for the prediction. And then, of course, also additional person-specific features from the blood tests and various questionnaires. And so integrating all of these features and applying this leave one out cross-validation scheme, we could significantly increase the prediction quality to 0.68. And you can see doing much better than the state of the art. And then after concluding this predictor, we actually wanted to put it to a more challenging test. So we collected a new cohort of 100 people, independent cohort, and then we applied the algorithm developed on the initial 800 people in the training data to this validation cohort, and we were very happy to see that the quality of these predictions were as good as those on the original training data. We could also introspect the algorithm and see, just get some insights into what are actually the features that are involved in the prediction. Not surprisingly, the single most dominant feature was the amount of carbohydrates in the meal, that the higher they are, the higher the predicted response is. Interestingly, though, the more fat in the meal, the lower the predicted response is. Then various other features related to the meal were also playing a role. And interestingly, many and dozens of different features coming from the microbiome, each making a small contribution to the prediction, but overall, all of them together making a substantial contribution, were also involved in this task. So at that stage, we wanted to ask whether we can actually use these predictions to design personally tailored diets for individuals and see whether by that we can actually normalize and improve blood glucose response. And so we designed a new study involving 26 new participants that we recruited into a double-blinded study where we profiled them as we did for the main cohort, and then we asked the algorithm to predict for each of them what we call a good diet that is supposed to elicit normal blood glucose levels, and a bad diet supposed to elicit very large spikes. And by design, the diets actually had to be identical in calories. In fact, every meal had to be the same in calories. Now, to show you that the diets are not trivial, oh, this stopped working. Oh, here. Oh, okay, that ruined my story. But I was going to ask you to predict actually which diet is the good one and which is the bad one, which actually when we did this in my own group, the students who developed the algorithm actually were split 50-50. And since I ruined it already, I'll show you that here, this is the diet that was actually predicted to be the good one. But now let me show you what these diets actually did in the individual. And this individual is, by the way, pre-diabetic. And I'll show you in the next slide the continuous blood glucose levels when following these two diets that, as I mentioned, are equal in calories. And what you can see is in the same individual, in the red is the bad diet. You can see that the red diet really elicits spikes that reach abnormal levels, indicating that this participant is pre-diabetic. But this same participant on a diet with the same amount of calories that, as I showed you on the previous slide, even contained ice cream, this same pre-diabetic participant achieves fully normalized blood glucose levels across the entire week without even a single spike in blood glucose levels. And here's, just for fun, another diet of a different participant. See if you can guess for yourself which one is the good diet and which one is the bad diet. For this participant, this one is the good diet, the one, yeah, with the croissant and the hummus. Hummus coming from Israel, of course, has to be on the good diet. And this, again, qualitatively, very similar results. And in fact, in most participants, we found highly significant differences in the good diet compared to the bad diet. And just to show you the personalized nature of these diets, you can see here that there's some foods that were given to some participants on their bad diet, but to other participants on their good diet week. And so just to, and I'll finally very briefly just mention that we've also tracked the microbiome during these short-term dietary interventions, and we actually found that these elicited significant changes in gut bacteria, some of them being consistent across study participants. And for all that we could tell, according to what's known in the literature, these bacterial changes induced by the good diet were largely beneficial because bacteria that in other studies were associated with good outcomes tended to increase consistently following the good diet, and bacteria associated with disease in the literature tended to decrease during the good diet. So just to summarize this story, I told you about work that we did on trying to understand healthy nutrition, looking at blood glucose levels as one healthy marker. I showed you that blood glucose levels after meals are highly personalized, highly variable across people, and that a machine learning algorithm integrating multiple clinical data and gut bacteria features can accurately predict these differences, and that we can take those outcomes and design personalized diets that normalize blood glucose levels in individuals. And in the last 5 minutes, I want to tell you very briefly about a news story that we've been working on, asking the question of why it's so difficult to maintain weight after a diet. And many of us actually know this phenomena. It's relatively easier to lose weight on a diet on a short term, but then typically you can gain that weight back, and that's what's been termed the cycling effect or the yo-yo effect of dieting. And so we wanted to study that, and we designed an animal mouse model where on one extreme we have mice on a normal diet, and on the other extreme we have mice on a high-fat diet. And then we also introduced this cycling where we feed the mice a high-fat diet, and then in various different ways, we make the mice lose weight, and at that point, we give them again the high-fat diet. And indeed, in this animal model, we could recapitulate what is known as in human, that actually, if you take mice that didn't have this history of obesity, and you give them the high-fat diet at the same point, which we'll call the nadir point, then at that point, you get less weight gain by these mice. And these differences are significant both in weight and in other metabolic derangements that I don't have time to show. And so this really suggests that somewhere there's this encoding of a memory of previous obesity in this point where the two groups of mice that will see the second high-fat diet challenge actually have equal weight. And to make a long story short, we looked at many different metabolic parameters, and we could find no differences between these red and green groups of mice that had the same weight at this point, except in their gut bacteria composition. So when we look at these at the gut bacteria across this timeline, when we start out, and this is a PCA plot of the high-dimensional gut bacterial compositional space, all of these groups of mice, as you would expect, are kind of mixed. On a high-fat diet, as has been documented previously in the literature, we see that on a high-fat diet there's a shift in the gut bacteria. But interestingly, even after this group of red mice is brought down to the same weight as the black group of mice, this change in the gut bacteria can still be detected. Okay, but this is, of course, associative. The question is whether this has any causal role. And this is where it's really, we can use the power of having this in an animal mouse model and really prove that the gut bacteria are actually the ones causing this enhanced weight regain after the diet. And we've shown this in two different ways. One is if we prescribe antibiotics at the point where the two mice groups are equilibrated in their weight, then we actually abolish this enhanced weight gain, and we also see an equilibration of the gut bacterial composition. And a second way by which we can do this is by microbiome transplantation experiments, where we take from this point where the two groups of mice have equal weight, we take their gut bacteria and we transfer them into germ-free mice, and we can see that by that we can recapitulate the effect such that in germ-free mice, those mice that received the bacteria from the group of mice that actually had the previous history of obesity, they gain more weight on the same diet compared to germ-free mice that receive bacteria from mice that didn't previously have a history of obesity. So we can transfer this phenotype simply by transferring the gut bacteria. And then, since the bacteria play a causal role, we also ask whether we can use their composition to actually predict what would be the exact weight regain on a high-fat diet. And indeed, we found that using only the gut bacteria population at the point of equal weight, we could also not only distinguish between the two groups, but in fact predict the exact weight regain on a high-fat diet in each individual mice with fairly high accuracy. And when introspecting the model here, we could see that it wasn't just a single bacteria, but in fact the entire composition of the bacterial population with multiple different bacteria that were the ones contributing to the overall predictions. And then finally in this story, if the gut bacteria have a causal role in this phenomena, we asked whether we could also intervene by intervening in the gut bacteria to maybe ameliorate this condition. And this we also did in two different ways. One is if you actually do a microbiome transplant in the gut bacterial population at the point of equal weight, then you can actually abolish this enhanced weight regain. And a second way is that we've also done an extensive metabolomics profile at the point where the two groups of mice had equal weight. And here too, we also found that there were several metabolites that were actually significantly different, in particular, two metabolites involved in the flavonoid biosynthesis pathway that were significantly reduced in the mice that had a previous history of obesity. And reintroducing these metabolites to the mice with previous history of obesity actually also ameliorated the condition and reduced the enhanced weight regain. So with that, I'll just wrap up this story studying cycling obesity, where I showed you that the microbiome has a causal role in this enhanced weight regain after a diet, that we can then use this bacterial population to, at that point, to in fact predict quite accurately what would be the weight of regain in mice, and again also in mice, at least offer two different ways of intervening directly with the gut bacterial population to ameliorate this condition. And with that, let me just end and say that all of these projects I mentioned were done in close collaboration with my friend and colleague from the Weizmann Institute, Eran Elinav, and many different graduate students and postdocs that greatly contributed and led this entire work. And thank you for your attention.
I'm going to jump in with the first and most obvious question, which is, if I like pizza and it's not meant for me, can I somehow causally use someone's microbiome for whom it is good, such that I can eat pizza?
Yeah.
And are you exploring this? I think it could be a big money maker.
Yeah, so good question. I think we're still far from that for two reasons. In the first project, what I showed is that we can actually use multiple different parameters to predict somebody's response, and prediction is not causation in that sense. So that's one way. And the second is that still, yeah. So I think we still need to really figure out the causal role, and then separate from that, how do you actually intervene in a bacterial population in a provably predictable way to alter it. So I think we're still far, but actually I think we can even using the gut bacteria and the other clinical parameters to make predictions, that I think is actually a reality that we can actually use today to tell people what may be better food choices for them in order to achieve blood glucose normalization.
I have a question over here. Thank you for the interesting presentation, first of all. I enjoyed following it. Your graphs, or rather slides, that showed good versus bad diet, triggered my attention. So from my extensive nutrition fitness experience, I remember learning and reading that, typically diets that are rich in carbohydrates would cause spikes in glucose fluid levels. So therefore, your first example of bad versus good diet was not surprising. Now, the second one, which had a good diet, which comprised rice and goulash, was somewhat surprising, but still on a second thought, I know also that complex polyhydrates like starch and fiber, they cause increase in glucose level, but not spikes, okay? Are there any insights that you can share, in particular regarding the second diet, beyond this clustering that you showed into good and bad diets?
Yeah.
Thank you again.
So these are all very good points, and let me again point out, as I mentioned in the talk, so the carbohydrates is actually a significant feature in our predictor, as it should be, because it's really highly correlated. So the overall algorithm actually makes use of the amount of carbohydrates, and that's definitely a prominent feature. However, I also showed on our data what is the predictive power of carbohydrates. It's highly significant, but it's actually quite weak. And so what the algorithm does by integrating these many different other features is actually improves the prediction much beyond that. So in other words, what rice or pizza or something that is richer in carbohydrates, while on average will probably elicit some response in some people, the response will really still be variable, and that by itself only explains some fraction or 15% of the variation, whereas the overall algorithm can reach 50% of the variation.
And that probably goes along the classification in the second case between good diet and bad diet, how these different food items were.
Yeah, and as I mentioned, some foods that were, it really is the case that in the diets we prescribed, some foods that were given to some people on what we call their good diet were really given to others on their bad diet. So there's even extreme cases like that. I would say that it's more the case that less that you have something that's good for somebody is bad for somebody else, but something that's good for somebody may be moderate for somebody else. And
So if I may also give you last comment or other feedback maybe for follow-up studies. So there have been diets proposed in the past where people looked at decreasing considerably your carbohydrates level, and at the point at which your body will start burning fat, so that it produces energy. Is there a way of integrating that knowledge or insights from those studies into what you have been doing here for even better performance? Or?
Yeah, no, that could be something interesting to do, which we haven't done, and one has to think of how to even measure that.
Great. I think we have time for just one more question. I'm going to let Donna take it.
So first, a short comment. I want your microbiome because we once told me you could have all the chocolate ice cream you want, and I've hated you ever since. But the more concrete question, other than declaring my hate and jealousy, is that you said that with the mouse experiments, giving antibiotics erased the weight memory. And unfortunately, from personal experience, that doesn't cut it in humans. So what other factor do you think is preserved that the antibiotics aren't erasing in us? Or you just think you take enough antibiotics and maybe I should overdose on that?
No, no. So I think the antibiotics was just it's a proof of principle. I mean, it's one way to show that the microbiome has a causal effect, and it's not that we're saying and that we think that really that should be a way in humans to treat that condition, because obviously antibiotics, and this has also been documented, have many detrimental other effects. So what else is it? I mean, I think it's complex. I can tell you that mechanistically, the place where we're seeking insights, and I've also shown results on that, is in the metabolomics. So looking at actually the metabolites that are actually different in these different conditions, at least in the mouse, allowed us to identify some metabolites that when you restore them also ameliorated the conditions. And metabolites are really the level that interfaces, that's the interface really between between the host or us and the gut bacteria. So that's the next place where we're actually doing this, and now we're looking into doing that in humans and trying to identify that, where it's actually a much more complex system, obviously, than these genetically identical mice that we control their habitat.
Great. Let's thank the speakers one more time. And also, I'd like to thank my co-organizers, Jonathan Carlson, Hoifung Poon, and Nicolo, for this great session. Thanks.